just okay, I'm just gonna share.
Yeah, no worries. You can hear you. It's just a little bit distracted, but I'm sure our ears will get used to that pretty soon.
Yeah. We have a good comment from Yen Yi saying that maybe if you speak slower, that would help. Just make sure everything you're saying is pretty clear.
Yeah, no problems.
And
So,
yeah, I think we'll be ready to start. So I'll just do a little introduction because I have so many nice things to say about this speaker today. I worked with Carol for several years at Purdue University, and this guy has studied some very interesting things, from the sounds that different beetle species make within tree trunks, like the vibrations, to detecting rainfall, to frogs, and now with detection of individual birds. He's one of the most computationally savvy and with a real thirst and love for the scientific research, reading papers all the time. And it's great to learn what he's been up to, living in New Zealand for now it's like 4 or 5 years, right?
7.
7? Jesus.
Yeah.
Okay. So
Thanks,
yeah,
guys.
I'm very happy to welcome Carol Bedoya, and floor is yours.
Yeah, thank you. So yeah, as Ben just said, my name is Carl Bedoya. I work for Ataro Sanctuary, which is a small privately funded bioacoustics research lab. And today I'm going to show the work that we are doing on acoustic censusing and individual identification of birds. And the work we're doing is mostly focused on animal conservation. We really want to develop and give tools that helps communities to monitor species and improve their outcomes related to conservation. So yeah, when using computational bioacoustics for monitoring biodiversity, usually label data to hierarchical levels. The first one is species, the second one individual. Depending on the kind of data you have, you can answer different kind of questions at different ecological levels. For example, at the community level, you would be interested in knowing how many species are in a site, which is the number of species per unit of area, which is the spatial distribution of those of some of those species. At the individual level or at the population level, you would be interested in some questions that are related but actually kind of different. For instance, how many individuals are there in a site, which of those individuals weren't there before, which of those individuals died, which is the territory of some individuals. At some point, you can even combine information from those two hierarchies and ask questions related to the illness. So that is which is the abundance of individuals per species, or estimating some sort of biodiversity metric such as the biodiversity index that we know, which is just the number of species divided by the number of individuals. The thing is that bioacoustic research is heavily biased towards the species side of the equation. So I have two hypotheses for why that is happening. The first one is that data collection for this kind of problem is tedious, is really hard. You have to go to the field, you have to catch an individual, you have to tag an individual, then you have to release it. And after that, you have to follow it for a microphone and probably for several days or years. So it's very, very tedious. And the second one is that the initial methods that were developed for this, they were actually very kind of accurate, not very accurate, but the accuracies were decent. The thing is that the problem was that they were extremely over-engineered for a single species. So when you wanted to generalize that approach, you just practically had to develop a new method. Fortunately, I just feel that's not a limitation anymore because of the new deep learning methods that we currently have. So I feel those two things are not that complicated as they used to be anymore. And so the motivation for us is that firstly, there is a gap in the literature that needs to be filled related to individual identification and to the monitoring of animal populations. And the second one is some sort of dissatisfaction related with the way that gap is getting currently filled. A common approach that I've seen is that usually record the specific sounds of a site and you estimate a time frequency representation from that site. That could be the spectrogram. And then from that spectrogram, you estimate some sort of scalar value. That we call it an index. That scalar value is based on the forward distribution of the signal in a specific frequency band or frequency bands. Then you basically correlate that index against some in situ observations that you perform at the same time the recordings were collected, and then you generate some sort of model that correlates those indexes with the number of individuals. The problem here is that a lot of information is lost during the aggregation process for the estimation of the index. And I don't see how a problem that complicated, such as the censusing of animal populations, could be solved without such loss of information. The second thing is that during the aggregation process, you are probably adding some sources that are not related to the species of interest, which might make the metric sensitive to other type of sounds. And the third one is that you cannot estimate which are the individual sources that contributed to that emergent behavior that you are measuring. So it leaves you with that dissatisfaction in your mouth. So a possible solution to this problem is, or the way we are approaching this is first to characterize vocal individuality and then use that vocal individuality to study animal populations. That way we can use it to census how many individuals are in a site, to determine which of those individuals are new, which ones died, and which are the territories of those specific individuals and track them across time. We can also use them as an equivalent of the physical tags that we currently use. So this is actually a passive way of monitoring individuals. So I'm just going to give some background before I can disclose actually how we are doing this. If you have worked in the area for a while, you have noticed that the field dramatically changed in around 2016, and the trigger of that change was deep learning. After that, basically all methods of the state of the art methods that are dominating all the competitions and all the issues that we currently have are deep learning based, and they are incredibly cool. I like them a lot, and probably the coolest feature they have, in my opinion, this is probably one of the most basic architecture of a convolutional neural network, is that they integrate the feature extraction and the classification part of a pattern recognition process in the same workflow. This simplifies things are made because you don't have to manually create a set of features for a specific problem. We make things a lot faster and also makes your method easier to generalize. But one of the main issues of these kind of approaches is that they are very powerful, but they are supervised in nature. That is, you have to know which are the things that you want to categorize. That is the number of classes of the problem, and you need examples for each of those classes. So, what if you have, for example, novel elements that were not in the initial training set as it is common in ecological research. You just have individuals that pop up out of nowhere in a territory. Or even more, what if you don't even know what is there? What if you don't even know what's the number of classes for the problem, as it is the issue that we have with the censusing. So,
Hey, Carol,
yeah.
I have an idea. So we've recorded this presentation. It's sort of tough to hear some of the words that you're saying. Why don't we do this? If you can record a voice memo on your phone at the same time, I'll go in and edit the video with the good audio. Do you have a cell phone near you that you can do a voice memo with?
I actually have it. So yeah, I can. Just give me 1 second.
Sure.
I thought it was just me.
No, maybe the mic on the computer might be,
It's supposed for me, and I could do it another day. But no, I'm just gonna record it.
You also can join through your cell phone if possible, and then you can just use your cell phone mic for the audio. And,
that's a great idea.
That's actually genius. Thank you.
What a genius. Who was that? Can we give a shout out?
Nice. Yeah.
I was a genius.
Please introduce yourself. Thank you.
Thank you so much.
I hope there's going to be a feedback.
Yeah, I hope so. Let's try. Yeah, it's going.
Oh, what an idea. This just goes to show you what a community of PAM practitioners can do with that 1 or 2 researchers talking to each other can't.
So many of the time these things happens to our students as well. So we sometimes have solutions like if students give the presentation to somebody else, they just put the audio due to the poor network connection. This all the alteration we do during this the presentations.
Carol, I just WhatsApped you the Zoom link.
Oh yeah, I just opened it.
We really did it. I was so bummed that the sound would be difficult quality, and now it's just the perfect time.
Can you hear?
And Carol, you also have to mute your speaker as well as the mic in your computer. Otherwise, you will feel an echo.
Yeah.
You have to mute your speaker as well as the mic.
Yeah, what about that? I can hear myself. So yeah, I'm just gonna, can you please talk?
Can I talk? I could talk forever.
Yeah. No, that's awesome. Do you receive some feedback or not?
No.
Okay, cool. That's perfect. Do you want me to repeat that? No, let's just continue from here.
Yeah, maybe it's at the start of this slide, because I think the audio got a little worse. But just turn your screen on so that we can continue to, oh no, I guess that's, yeah, everything's working great.
Okay, perfect. So yeah,
Carol?
so you have this set of techniques that are very powerful, but that are supervised in nature. So what we can to tackle, if we want to tackle a different kind of issue with those, such as identifying new elements that weren't in the training data set, such as individuals that were not initially in the territory in which we were performing the monitoring, or what if we don't even know which individuals were there or how many were there. So basically, our challenge is to take advantage of these very powerful deep learning techniques and apply them to an inherently unsupervised task, or in this case, censusing. So this has a name in bioacoustic research. The specific two problems that we are tackling are called, the first one, open set recognition, which is when you have several known classes and many unknown classes. And the second one is unsupervised learning, which happens when you have no labels available and you don't know what is there. The number of classes is unknown. These two kind of problems and many problems have been discussed in detail by Stowell in this paper. So in case you want to read it, I feel it's really cool. It gives a general introduction to the area and the problems that are left to tackle. So there are too many ways of addressing the problem that I'm just stating. I'm just going to show you the approach that I selected, which I consider is the best based on my limited experience and background. There could be like many others that are equally as valuable. So there are these two papers that basically summarize the approaches used to tackle every kind of problem. I selected some deep clustering based because technically I can tackle both the unseen individual identification and the censusing of the birds using just a single approach. So in general, these kind of methods work in this way. You have some raw data, then you use a neural network to estimate some low dimensional representation of that data. That kind of neural network could be like a classic convolutional neural network. It could be an autoencoder. It could be a generative model, like a parational autoencoder of a GAN. Then you use that representation and perform clustering on that representation, and then you get some sort assignment from that clustering that is some fuzzy or probabilistic distribution for each element. Probably the most common way of determining that representation is using autoencoders. This is the typical structure for that. You get the data, you just encode it. An autoencoder is a neural network that gives you, that tries to replicate the input. So why do you do that? What you do is that you put the data through a bottleneck, trying to reduce the dimensionality on the data. Then you perform a clustering at the end of that bottleneck, and then you combine both clustering and you combine the losses of the clustering algorithm and the neural network together, and then you perform the training together. This is like a typical example of that network applied in what do you call that? In another problem. So you just get the data, you pass it through an autoencoder, and in this case they use k-means, and you can see this is basically that. So this is probably the most common structure that you would see for these problems. This
is actually getting widely used in molecular biology. We are really living the golden age of the clustering. Papers like this have been popping up in Nature left and right during the last 2 years. So seeing that this was so common in Nature, and it was solving like problems that were in paper more difficult than the one we're currently tackling, I was wondering if they were being used in bioacoustics. So I just reviewed the literature and I found one, which was very surprising to me. In this case, what the author do is applying deep embedded clustering, which is like probably the initial technique in deep clustering. In a two-class classification problem, they just wanted to tell apart whale calls from fish towns, and they found that they get practically like the same accuracy as the k-means, but without the need of manually curating a set of features. This is not the kind of problem I would use clustering because they only have 2 classes, but it's pretty cool that people is starting to use this kind of approaches. I also found some papers that are tangentially related to this issue, but they are not quite the same. This is the only one that I've seen that properly applies these kind of techniques. So in our case, how does our approach work? So typically when you want to perform individual identification, you just get some sort of an input. In this case, this is an analogy. You get a panda, you pass that panda through a neural network, and at the end, you just give your result. So yeah, it tells you that's a panda. So what we do is that we don't use this neural network as a classifier, but we just mostly focus on the feature extraction part. So we use it as a feature extractor, and then the clustering algorithm is the one in charge of determining if that panda, if that is a panda or not. The thing is that as I showed you previously, those neural networks usually have 2 stages, one dedicated to feature extraction, the other one dedicated to classification. And the problem is that the classification part carries a lot of weight. Therefore, the features that the network usually finds for classification are not exactly the same that are needed for clustering. So a way of like compensating this balance is just to perform a joint training between the clustering algorithm and the network and the neural network to solve that issue. So when you get like a new data, you just extract features from that data, and then you use the clustering algorithm, and the clustering algorithm asks, is this the class that you want to, is this class? And if that's the class, it just classifies it in a box. And if not, it just creates a new class with the parameters of that new element. So this is in more detail what we are currently doing. So we have a vocalization of a pair, in this case a Kiwi. Then we augment that data with background noise of the environment. Then we apply some sort of median filter to, then we estimate the time frequency representation, the spectrogram, applying some median filter to remove noises that are actually quite easy to remove. And then we use that as input to a convolutional neural network. So that structure there is enough to identify individuals. And I noticed that this is actually not the challenging part of the approach. When we tested several networks, the results were actually pretty good. And when you look at the literature, most of them follow that structure, and they seem to have no issue in that regard. I obviously modified this network a little bit. This was initially BGG19, which is an architecture. I just changed a bit, so I changed it a bit so I could reduce the latent space for the clustering algorithm, because when it's too big, most clustering algorithms have issue performing a clustering task when the number of dimensions is too high. Sorry if I confused you there. So yeah, then I get this latent space, which is the part that connects the feature extraction part from the classification part, and I use it as an input to a clustering algorithm. So, but this clustering algorithm is special. It doesn't work with any clustering algorithm. What's interesting about this one is that it does not require the number of classes as input parameter. So when you are going to use, for example, k-means, means, gk-means, the first thing they ask you is in how many clusters do you want to divide that data. But in this case, you don't need it, and that's what makes this architecture special. So in theory, it should work that way. So you just get the data, reduce the dimensionality, and apply the clustering, and that's it. But it actually doesn't work. So it happens that to solve the issue that I told you a few minutes ago, in order to give more weight to the features extraction part of the algorithm, you just have to train both together, the clustering and convolutional neural network. This is probably the most difficult part of implementing these approaches. And yeah, that pretty much solves the issue. So yeah, if you want to divide this general architecture in stages, you can just call it preprocessing, feature extraction, and clustering, which is performed jointly together. So why does this work with this clustering algorithm? This clustering algorithm differs from the ones that I just previously mentioned in that it is not based on the optimization of an objective function. Most clustering algorithms, it just works in the ways of these old fuzzy expert systems that used to be very popular in the 19s used to work. So what you get is that you get the input data, then you fuzzify that data, you just make it big, you perform some inference on that data, and then you just aggregate that information and get the outputs. So the difference with those expert systems is that this algorithm doesn't require that knowledge base. It just infers by itself. So when you see the structures is something like that, but if we divide it into stages, it's just this. You just get the inputs, you fuzzify them, perform inference, and diffusify. We use a particular version of this algorithm that we developed that naturally restricts the number of generated clusters. There was like a little theoretical issue with that used to produce a huge number of clusters that were not desired. So this is called the full reinforced version of Lambda, which is the one we currently use. So what makes this work? So to perform unseen individual discovery and to perform acoustic censusing, you just need two things. You need an accurate individual identifier, which is actually not that difficult to get if the species is not super vocally complex. And you need to perform joint training with a clustering algorithm that allows online learning, not any clustering algorithm. Online learning means that it analyzes data as it goes. Something that I forgot to tell you here is that as you see, this process is not iterative, so you just get the data and gives you a result in one go. So this is what makes it special. And then after that, you get like a basic accuracy, and then you perform some modifications on the strategy to get some marginal gains that increase the accuracy. You just add a cluster validation stage, augment the data. This actually increase the accuracy quite a bit. You notice reduce the signals. You utilize and you select a good network architecture. You have to perform some process there. As a case study, I'm just going to show what we have done with the great spotted kiwi, which is this is a cultural significance for New Zealand. It's quite important, and also individuals are very, very difficult to handle. So yeah, it's one of the species that you don't want to touch, and that's why we decided to use it as an example. So here, this is probably like my favorite plot. we compare the results of our approach against two, or probably the two most commonly used acoustic feature sets in bioacoustics. So in the first one, I have 7 call features, the ones that usually extract in any bioacoustic research. We have the number of syllables, the intersyllable interval, the call duration, the spectral centroid, the dominant frequency, the bandwidth. And we can see there is that when we use those features, we can pretty much tell apart male from females, but we cannot tell individuals apart. When we use the male frequency cepstral coefficients, which were some features designed to imitate the way the auditory system of humans work, we can see that some clusters start to develop, but the problem is not resolved quite yet. By the way, there are 30 individuals here. And in this approach, we just can see how clusters develop like so crispy and compact and separated so from each other. It actually makes these tags relatively easy. So yeah, it's embedding this performance is just awesome, and it's not surprising for me that it's used that much in molecular biology. So regarding individual identification, as I told you, you can use several of those pre-trained networks that are already there. They are widely available in Python or in MATLAB, and any of those will give you a decent accuracy. You can tune them a bit to increase it, or in this case, to solve the issue with the clustering. If the network is too deep and the latent space is too wide, you start having issues regularizing the network. So yeah, there is a little bit of tuning there. So yeah, we just choose the one that was more parsimonious for us. And yeah, if you want to perform individual ID, that's pretty much everything you want to do. For instance, individual discovery, we tested the network. We trained it with 10 males and 10 females, and then we added, then we wanted to recognize again those 10 males and 10 females, but we added 3 individuals, 3 males and 3 females that weren't there, and we can see that we can detect the clusters of the individuals that weren't there, and even we can relate each sound to each of these clusters, which is fascinating to me. So it not only gives you the exact number of things that are new, but also which calls belongs to those things. Similarly, for censusing, we just added 10 individuals, 6 males and 4 females that weren't there, and we can see how we can cluster with a relatively good accuracy, 88%. How these clusters get like tightly joined together, and we can get not only how many things are there, but some sort of relationship between the things that sing and the class that is detected, which is amazing to me. It really like blows my mind. We currently have a paper on the topic. We have the preprint. We also give away the data and the code in MATLAB for the people that want to use it. But something that I want to say is that we perform like significant modifications on this preprint. Those modifications is that we extended the number of pairs from 1 to 10 because we really want to show the capabilities of our approach, and I feel that only one species was making it relatively weak. And the second one is that we moved everything from MATLAB to Python. Actually, it's currently coded in PyTorch because MATLAB is super expensive, and for us it's not an issue, but most people don't have the resources to pay for MATLAB. So we just decided that if we wanted this to be used, we just have to use an open framework. So yeah, we're making those two things. I'll upload it probably around the end of October, once I come back from vacations, and yeah. We also have a visualization tool in case you want to play with our results. This is literally power, but that method that I just showed to you. Here you can see which individual is vocalizing and where, and then you have a way of visualizing and hearing the vocalization of each individual detected. So you cannot like only see what it is, you can also compare it, the results are good and everything. It's quite a good, interesting tool, but it's mostly educational. I would like to acknowledge all people that helped us to do this, because data collection of this is ridiculously hard, and also to the parent group that supported us on this. Yeah, that's pretty much it. These are our emails in case you want to contact us. Thanks so very much.
Carol, that was really awesome. We've got a
Thank
few questions in the chat.
Yeah.
Let's see. So Yenyi, are you here?
Yes, I'm here. Hi.
Hi. Would you like to either read your question or just say it again?
Yeah. Yeah. So in one of your slides, you mentioned the steps for the censusing to work. And I wondered, for the number one, it was accurate individual identifier. So yeah, I wondered how different do individuals need to be in order for the clustering to work?
Yeah, that's actually a pretty cool question. There are like different ways of characterizing individuality. So one of them is mostly related to morphological changes in the individual. So it's like the pitch is higher, the duration is wider. The other one are individuality related to behavioral differences. So like, yeah, they have like different sequences and all like that. This one is mostly designed to detect those small differences in those morphological differences, like the pitch is a bit higher, the duration is a bit wider. So yeah, how different do they have to be? So in my opinion, it's like an in-between. They have to be somehow different, but not that different, like at the species level. So yeah, that's probably like the best answer. It's something in between. Yeah, it's something in between the same call and the difference that you get at the species level. So yeah, so that's one thing. And the other thing is that you also need like a relatively good amount of examples, like around like between 50 and 200. So the network can actually like discern those features. But that's actually like the cool thing about these approaches is that that you don't have to curate them. So what I'm gonna do to approach that is like once I disclose like the results for all 10 species, I feel I be able to give some sort of metric of differences, and so I could actually like quantitatively answer the question, but it's really good.
Cool, thank you.
Yeah, no
because that's just assuming if the species do have vocal signature between individuals, right? Like, I think my question is also related to Fernando's question on, yeah, inter-individual variation and stuff like that. So maybe you can answer that next.
Yeah. I'll go with that one next.
So Fernando is asking, for vocal individuality, at least two requirements are required: high inter-individual variation, and that such variation remains constant over time. How did your research account for such variation over time?
Yeah, that's actually a very good question. you have two things. One is vocal individuality, which is some sort of pattern that stays over time, and the other one is discrimination, which means separating individuals at the same time. So in order to perform that, you just need individuals to sing differently at that thing. So with Kiwi, we didn't approach that because we don't have calls that are like from different years. But for the other species, we have that. We have a species that individuals that were collected at different times across years, and with those species, the acoustic signature stays there. It's like the latent space that you extract from those recordings that were collected at different station, different time, preserved. So yeah, for Kiwi, no. For Kiwi, I would say this is mostly discrimination more than individual identification, but for the other birds, definitely that sustains.
Rouge.
Yeah, hi. Thank you so much for this interesting talk. So I had this question in the beginning, but I think along the like the rest of the slides, I kind of have an idea now of why you used clustering instead of using classification, because I think you're not, so your number of classes is not fixed, right? When you start, and it keeps on changing. But now I have a different question here, and that's more about the method. So when you kind of have a feedback mechanism where you train you start off with your labels, and with these labels, you then try to train your parameters, like hyperparameters of your network, and learn better and better features. And you do it in a way as your clustering gets finer, right? You get more clusters. But when it comes to later, when you use data that the network hasn't seen, does it just create a new cluster, median points or mean points, or so what does it do? Does it do any learning at that point? Because now you're not learning, right? I'm talking about the part when you're doing the inference.
Yeah. That's actually, that's awesome. Yeah, these people is very smart. So yeah, what we do is that we don't move the parameters of the feature extraction part of the network, but actually the parameters of the clustering are updated. So yeah, the network part stays fixed, and then the cluster keeps updating the cluster prototypes. So yeah, that's what happens.
Okay, so basically what you're saying is that you learn the features so you train the network based on your data, which gives you your latent space, and then you feed it to generate your clusters. But when you get a new data sample, like for instance, you get a new recording, you do run it through the convolutional neural network first to get your features, right? And these features are still you still get it from the same network.
So yeah, the thing is that something that I would like to clarify there is that everything is not performed independently. So at the very beginning, you just train the network and the clustering part, and then once everything is trained, you just disconnect them. You just, that link that connects the cluster and the network, you just like separate them, and you just use the feature extraction part of the network. So it would go like input, feature extraction, no classification, no connection to clustering, then you go to clustering. So when you get something that is not in the initial number of classes, so the cluster prototypes, which is basically what determines which individuals is there, these ones get updated, but the feature extraction just stay the same. So it's like after that, the weight is carried by the clustering algorithm, not by the feature extraction part.
But the number of clusters can still change, right?
Yeah, it can change.
But only based on the new features now, or the new set of data that you have.
Yeah. So yeah, definitely, yeah.
All right.
Yeah,
And
that was
what
happening.
kind- okay.
But that's for that case, the unseen individual discovery. Because when you have censusing, that's pretty much the result.
Right.
Just cluster the space and that's the result.
Okay, but does the clustering happen in an unsupervised way?
Yeah.
Just the clustering part. Just the clustering part.
Yeah.
So the clustering happens in an unsupervised way.
Yeah. That's actually that the first time I saw that, that's like, wow, this is impressive, because the algorithm not only optimizes the result, but also the number of classes.
Right.
So yeah, that's actually very impressive. Just try it.
Yeah. So the clustering happens in an unsupervised way, but still the number of clusters that you get, you compare them with the annotated labels.
The thing is that you are like asking me about two different problems in the. So there are like two ways of doing this. So it's like the architecture is exactly the same, but depending on the problem, you use it in different ways. So if you want to perform censusing, you're just using like in an unsupervised way. But if you want to detect new individuals, you have to have some way of collecting that information of the known individuals there, because you need them. So there are like two cases. One case in which you want to perform monitoring and find some things that are there just in case. And the other one in which you have no idea what's in there and you want to know what's in there. So in one case, you need to use the cluster prototypes as input for the clustering algorithm. So yeah, in that case, this is this is like some sort of semi-supervision, because you just give like the cluster prototypes. But for the other one, you just let it unsupervised.
Okay. So how I understand it now is when you're training your network, you're changing your clusters based upon changing your features based on the results that you get from your clustering, right? And based on that, you're training your neural network, which gives you these features, like
they are combined together. It happens that if you disconnect, it's like, because another strategy, it could be just like input, you connect the network, but you just don't use the gain, like the loss from the network, but just the loss of the clustering algorithm. That's like a how to say, like a feasible approach. The thing is that if you don't connect the network, the clustering destroys, collapses the initial feature embedding. So, and it actually doesn't work. So yeah, it has to be there. The network has to be there during the training.
All right. Yeah, thank you so much for that. That clarifies it. Thanks.
Yeah, no worries.
Nice. Carol, one question from Amandine. Hi, Amandine. Is there an official name for this method that mixes deep learning and clustering?
I mean, the general frameworks are called deep clustering. So yeah, and there are like a lot of names there, deep embedded clustering. I think I just named it. I forgot the name, I just put it there, because this is not an application of a method, it's just a method in itself that falls into a family of deep clustering techniques. And to be honest, I don't remember the name, deep convolutional clustering network, something like that. I don't remember. But yeah, if you want to read about the topic in general, it's called deep clustering.
So one question that I have before we get to Micheline's question. So we have people here that are studying wolves, that are studying gibbons, just let's say animals that could be good candidates for this type of method. What would be the sort of steps, both with regards to data collection and labeling, and then on the analysis side that one would need to do in order to put this method into practice, and is hopefully potentially using some of the code that you're going to make available, code and other resources?
So definitely, probably like my main goal is that people uses this because yeah, I just like sharing in general. So yeah, I'm just based on some feedback I received. I'm just gonna make this as easy as I can. So the person, yeah, regarding the researcher, the first thing they have to do, probably the main thing, is that they have to guarantee that the sound that they are collecting belongs to that specific individual. So you probably would like to tag that individual or have unequivocally evidence that the individual that you are recording is actually the individual. So that's probably the main thing. Something after that, you just put the calls of every single individual in one folder, blah, 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 and that's it. Then you just run it, and at the end, you get like an Excel spreadsheet. That's pretty much it. The thing is, something that I currently don't like about my method is that the entire process is not end to end. You can notice that the segmentation stage wasn't there. So I perform segment, and actually I'm just gonna, with that code, I'm just gonna give a segmenter for that kind of data, like in which people can just put some example of the vocalizations, and then the segmenter can segment the rest. But I would like the technique to be completely end to end. You just put the recording, and then it performs the segmentation, the feature extraction, the classification, and the clustering and everything. It's just like a lot of work, and we are on the process. So yeah, so what do you need? You need like correctly and unequivocal evidence that the individual that you are collecting is that individual. Then you put calls of that individual in one folder. And so folder 1, individual 1, 50 calls, for example. Folder 2, individual 2, 100 calls. And you just let it run, and that's it.
Wow. And so you'll let it run on new data that's in a different folder.
You just let it run. No, I'm trying to put everything, so it makes like the splits. So yeah, you just get like the split for the training, you get the split for the validation, so people don't have to do that. So it performs the splits, the data augmentation, and yeah. So yeah, just trying to make it like as easy as possible for the people. Especially something that I've noticed is that a lot of the methods that we use cannot be used by people that perform some sort of activities related to animal conservation, especially something that is as easy as us as selecting, I don't know, the type of window for the Fourier transform or things like that are very difficult for someone that is not trained in the area. So I just want to lower the level a bit on that regard. I want obviously to give the researchers, the capabilities to change anything they want, but I also want to make this as easy to use as possible.
I think it's going to make a big impact when all of this becomes available. Micheline has a question also about the input features that you're using for classification, or if you're just giving more raw data. So what's that reduction stage like? Are you using spectrograms, indices? You may have mentioned that earlier.
I just input the general spectrogram, and that's it. Yeah, so yeah, the input for the framework is just an augmented noise reduced version of the spectrum, and that's it. Curiously, I feel that in few years we're gonna move away from that, because all the progress that is currently happening with recurrent neural networks, that they automatically generate the feature representation that they feel it's optimal. So that's actually quite fascinating. I feel that at some point in the future we wouldn't even need to do that.
So just, what were you saying, raw audio files?
Yeah,
Or-
yeah. It probably, as it happened with the feature extraction part, in which they automatically select the features by themselves. In the future, they are just going to select the best representation that is optimal for the problem. So that was actually one of the key elements to solve this big issue on protein folding. So instead of giving some transformation of the data, they just created a framework that was end-to-end that generated that transformation inside the network. And now we have this huge impact in molecular biology, probably an upcoming Nobel Prize. And so, yeah, that's probably going to be the future in that regard.
Any other questions? Okay, Carol, I'm really happy we were able to figure out that audio issue. And no, what a cool topic, and your work is amazing. So we'll eagerly wait sometime, hopefully in October. This paper will come out, and these tools will become available, and just really, really cool stuff.
Yeah, thank you. We are just trying to make it like something that works. Let's see. At the end, is the scientific community the one that determines what works and what doesn't work. So yeah, I'm very happy to receive all the feedback.
Great.
Mhm.
Take care.
Awesome.
Bye,
Yeah,
everybody.
bye.
See you in 2 weeks.
Yeah. See you. Bye.